I'm going to talk about complement system regulation, and the aim of complement system regulation is to protect host cells. That is to say, to restrict the activity of the complement system to non-self only, like bacteria and viruses and other microorganisms. So there are several factors that regulate the complement system. Some of them work on alternative pathway of the complement. For example, factor H, and as can be seen here, this is actually C3b, which is spontaneously cleaved and recognize non-self on the surface of self cell, and also recognize polysaccharides on self of microorganisms, and also recognize snake venom, so it will bind to this non-self antigen, and this will activate two factors, factor B and D, and factor B get cleaved by factor D into BA and BB. BB will bind, and this will lead to formation of C3 convertase, which act on C3, which is non-spontaneously cleaved and cleaved, and this will lead to formation of the MAC. Factor H, it will, as can be seen here, prevent C3b binding to factor B, and actually this is once the immune response is done, and the aim of this is to protect the host cell, and this is, as can be seen here, there is another factor which is known as factor I. As I mentioned, that factor B, it interferes with the binding of C3b to factor H. As I mentioned, factor H interferes with binding of C3b and factor B. However, factor I further cleaves C3b into bound IC3b fragment and soluble C3F fragments, and this is to prevent further activation of the complement pathway. Also, DAF, or decay accelerating factor, dissociate C4b from C2b when the immune response get done. And S protein prevent insertion of the MAC membrane attack complex. Actually, this is terminal of the MAC, which start from C5 to C9, and usually this terminal part, it removes phospholipids and lead to formation of holes or pores. So S protein prevents insertion of the terminal part of the membrane attack, just to save the nearby host cells. Similarly, C1 inhibitory factor, when the immune response is done, as can be seen here, this is immune complex. It's composed of C1, and this immune complex get bind to C1, and these are associated proteases, C1R, C1S, and these activate actually C4b, C4 and C2. So C1 inhibitory factor dissociate these proteases from this complex, so it will prevent further cleavage of C4 and C2 into complement fragments. And as can be seen here, there are in the classical pathway other proteins, for example C4b binding protein, and this interfere with binding of C4b to C2b. This is C4b C2b, so this will stop the further activation of the complement. And also in the classical pathway, the decay accelerating factor, it plays the same role by preventing C4b from binding to C2b. In case of Mannose-binding lectin pathway, which is important in defense or protection against some bacteria like Salmonella, Neisseria listeria, and in some other microorganisms, for example Candida albicans. So when the immune response is done, also C1 inhibitory factor and antithrombin are physiological inhibitor of Mannose-binding lectin pathway of the complement. Thank you very much, Professor Ahmed Bola.